Welcome, everyone. If you are a B.Pharm, Pharm.D, medical, nursing, or life science student prepping for those big university exams, GPAT or NIPER, you are in exactly the right place. Today's explainer is going to be your ultimate exam prep masterclass for understanding the rigorous and honestly fascinating world of clinical research. We're going to take all those complex, heavy concepts and break them down into simple, easily digestible frameworks that will actually stick in your memory long after exam day. So think about this for a second. How does a raw, untested chemical sitting in a laboratory beaker actually become a life-saving medicine on a hospital ward or a pharmacy shelf? Well, it's definitely not magic. Every single approved medicine you see out there has undergone years, sometimes decades, of meticulous testing to make absolutely sure it's safe and incredibly effective for human use. Today, we're going to reverse engineer that epic journey from the lab bench straight to the patient's bedside. Here is our game plan for today's explainer. We'll start with the basics of clinical research, then cover ethics and trial types, map out the drug development journey, break down the specific phases of clinical trials, and wrap up with the team and the trial process. All right, let's jump right into Section 1, Basics of Clinical Research. We'll start with some foundational definitions. People mix these two terms up all the time, but for your exams, you've got to remember this critical distinction. Clinical research is the broad umbrella. It covers everything from systematic observational studies to testing new surgical approaches, radiation therapies, or even just ways to improve a patient's overall quality of life. Clinical trials, on the other hand, they are just one specific form of clinical research. They're the highly controlled, systematic experimental studies specifically designed to evaluate the effectiveness and safety of a new medication, a biologic, or a medical device. Think of clinical research as the entire hospital and the clinical trial as one specific, highly secure experimental testing lab right inside it. So why do we actually go through all this trouble? Well, the fundamental purpose of all of this research is to increase our scientific knowledge about how well a diagnostic test or treatment actually works in a real specific patient population. It allows us to take a hypothesis, like, say, an observation that a certain plant extract might lower blood pressure, and systematically test it. Ultimately, it helps us generate hard scientific evidence to improve patient care, evaluate safety and efficacy, and really just make sense of the medical world around us. Moving on to Section 2, Ethics and Trial Types. Let's look at the rules of the game. Okay, grab your mental highlighters because you absolutely must memorize these 3 ethical pillars for your exams. First, respect for persons. This basically means treating every patient as an autonomous individual, which is exactly why we have such strict informed consent processes, so patients know exactly what they are signing up for. Second is beneficence. This is a really delicate balancing act where we try to maximize the potential good for society while minimizing the risk to the individual participant. And third, justice. This dictates that we treat all participants fairly, ensuring that the risks and the benefits of the research are equally shared across different demographics. Now, we categorize clinical trials in 2 main ways. First, by researcher behavior. Is it an observational study, where we're basically just monitoring a group over time? Or is it an interventional study, where we actively give a patient a new treatment to see what happens? The second way we categorize them is by their actual healthcare purpose. And this ranges from prevention and screening trials designed to catch diseases early, all the way down the line to treatment, quality of life, and compassionate use trials, which are typically for patients who have literally exhausted all other medical options. Let's hit Section 3, the drug development journey. From the lab, straight to the patient. The pipeline for a new drug is extensive, and I mean highly regulated. It kicks off with drug discovery and target identification in the lab. Then it moves into the preclinical phase, where drugs are tested in vitro, meaning cellular models, and in vivo, using animal subjects to check the basic properties. Now, only after a successful investigational new drug, or IND submission, does it cross over into the clinical development block. This right here is where human testing begins. If everything goes perfectly, it ends with the new drug application to regulatory authorities, and finally, post-marketing phase 4 surveillance. Which brings us to Section 4, Phases of Clinical Trials, the human testing stages. This is a fantastic cheat sheet for your notes. We start out with Phase 0, using a tiny group of maybe 10 to 15 subjects for human microdosing. It gives us very preliminary data on pharmacokinetics and pharmacodynamics, but really no solid data on safety or efficacy yet. Then we step up to Phase 1. This is the first major stage, using 20 to 100 healthy human volunteers. The primary goal here is not to cure a disease, it's simply to test safety, tolerability, and to figure out how high we can escalate the dose before side effects kick in. Once we know it's safe, boom, we move to phase 2. Now we're bringing in 20 to 300 actual patients who have the disease. This is where we finally test if the drug is effective. That's the exploratory efficacy, all while pinning down the optimal dose. Now, notice the huge shift as we scale up from phase 2 to phase 3. Phase 2 is what we call therapeutic exploratory. It's smaller, and we often split it into phase 2A just to assess dosing requirements and 2B to really look at efficacy. But phase 3 is a massive therapeutic confirmatory trial. We're talking anywhere from 300 to 3,000 subjects. This is where the new drug goes head-to-head. We are establishing its efficacy directly against the existing standard of care therapies to prove, once and for all, that it actually works better or is at least safer for a large patient population. But wait, the research doesn't just stop once a drug gets FDA or regulatory approval. Even after it hits the pharmacy shelves, phase 4 kicks in. These are our post-marketing studies, or PMS. The main goal here is long-term safety surveillance, otherwise known as pharmacovigilance, because we are now looking at hundreds of thousands of people taking this drug in real-world healthcare scenarios. We can finally determine its behavior in everyday life, monitor for extremely rare adverse effects, and evaluate exactly what happens if a patient misses a dose or takes way too much. Finally, Section 5, Team and Trial Process. Let's meet the people who make it all happen. When you walk into a clinical research site, you are going to see a very specific, well-oiled hierarchy. The principal investigator, or PI, is usually a doctor and is the ultimate responsible leader of the trial site. Next, you have the clinical research associate, or CRA. They act as the monitor appointed by the sponsor to verify data and make sure there is strict adherence to good clinical practice, or GCP. Then you have the clinical research coordinator, the CRC, who is essentially the backbone of the entire operation. They handle the critical administrative duties, recruit the patients, and manage the case report forms. And of course, they're all supported by data managers, statisticians, ethics committees, and regulatory bodies working tirelessly behind the scenes. So how does this whole team work together? The entire life cycle of a trial begins with developing and approving a very strict protocol. From there, investigators are selected and ethics approvals are processed. Next up, we hit the crucial stage of patient recruitment and the actual clinical trial participation. As the trial runs, massive amounts of data are collected, carefully reviewed, and filed away. Finally, the statisticians get their hands on it, analyze that data, and that leads directly to a comprehensive regulatory submission and the publication of the final report. Because human lives are quite literally on the line, clinical research is one of the most highly regulated industries on the planet. That means meticulous documentation is absolutely essential. The fundamental documents include the investigator's brochure, which holds all the clinical and non-clinical data. You've got the highly detailed protocol that acts as the study's ultimate rulebook, and the informed consent form to protect our patients. All the data is captured meticulously on case report forms, or CRFs. Without these documents and the official ethics committee approval letters, a trial simply cannot happen. In this industry, there's a saying you have to remember: if it isn't documented, it didn't happen. To bring all of this together, the absolutely crucial point you should take away from this explainer is this: clinical research ensures safety and effectiveness. It is the vital, highly regulated bridge that translates raw laboratory discoveries into the safe, scientifically backed treatments that actually improve patient outcomes in hospitals worldwide. Every single approved medicine you are going to dispense, prescribe, or administer in your future career is supported by this massive mountain of scientific evidence. Understanding these principles is an absolute must for every single healthcare professional. So I want to leave you with a question to ponder for your exams, and really for your future careers. Which of those 3 ethical principles, respect, beneficence, or justice, will you champion most as you step into the medical field? Think about it. If you found this explainer helpful for your exam prep, don't forget to like, share, and subscribe for more clinical research lectures and pharmacy breakdowns. Keep studying hard, and we'll see you in the next explainer.